So now we will read about the COVID-19, that is coronavirus disease 2019, that has been recently emerged and still ongoing. So this COVID-19 appeared in the Wuhan city of the China, and in the terminal stages of the 2019, and from then it is continuing till now. So this COVID-19 disease is caused by SARS-CoV-2 virus. SARS-CoV-2 virus. SARS-CoV-2 means severe acute respiratory syndrome coronavirus 2, which belongs to the family Coronaviridae, and subfamily is Coronavirinae, and there are several genera, alpha, beta, and so on. So, of which this SARS-CoV-2 belongs to the beta coronavirus genus. Okay, that belongs to the beta coronavirus genus. So, this SARS-CoV-2, this is not the only coronavirus that has affected the mankind. Rather, before this SARS-CoV-2, there have been multiple such epidemics by the coronavirus, like the other coronaviruses that have caused the epidemics are the SARS-CoV, severe acute respiratory syndrome coronavirus in China in 2003, that has caused the epidemic. MERS-CoV, which is the Middle East respiratory syndrome, that had affected the Middle East Asia in 2012, and this SARS-CoV-2 that is still ongoing, that has caused the worldwide pandemic from 2019. So earlier to SARS-CoV-2, there was SARS-CoV that has caused epidemic in 2003, and MERS-CoV that has caused the epidemic in 2012 in the Middle East Asia. Coming to the morphology of this SARS-CoV-2. Now, as we have discussed in the general virology, that morphology of any virus can be described in 4 points, that is the nucleic acid, the capsid, the symmetry, and the envelope. So SARS-CoV-2, as we have read, that this belongs to the Coronaviridae family, and that is a family which belongs to the, or that is a family that has those viruses which are enveloped and RNA viruses. So since it is enveloped, so envelope is present, lipoproteinous surrounding the nucleocapsid. So envelope is present, and also the nucleic acid is the RNA, because it is a RNA virus. So nucleic acid is RNA. In fact, these all SARS-CoV-2, MERS, this SARS-CoV-2, MERS, and uh, the SARS, MERS-CoV, and the SARS-CoV-2, all of them are enveloped RNA viruses. All of them are the enveloped RNA viruses, and they all belong to the Coronaviridae family. They all belong to the Coronaviridae family. Now, so nucleic acid is the RNA. The nucleic acid is the RNA, and when there is nucleic acid, the capsid is always present surrounding the nucleic acid, and the subunits are called as the capsomeres. Symmetry. If we talk about the symmetry, then the symmetry of this is helical symmetry, and as we have talked earlier, that the envelope is lipoproteinous in nature. The lipid part is derived from the host cell membrane, and protein part is virus coded. This we have already discussed in the general virology part. But the point to note here is that the proteins in the coronavirus structure are the, in the envelope of the coronavirus, are the spike protein, which is denoted with S, membrane protein denoted with M, and the envelope protein denoted with E. These proteins are very much important because according to these proteins, these, I mean, the genes which are coding for these proteins help us in detecting the virus. So this the genes which code these proteins, they are helpful for the diagnosis of the COVID-19, for the diagnosis of the COVID-19. Now coming to the pathogenesis of the COVID-19. So in pathogenesis, first we have to note the mode of transmission. So mode of transmission in case of the COVID-19 disease is mainly the respiratory droplets during that are released during coughing and sneezing. Then the contact transmission through surfaces and the fomites, and the aerosol transmission. So the transmission of the COVID-19 COVID I mean SARS-CoV-2 virus can occur through these three routes, that is the respiratory droplets, contact transmission, and the aerosol transmission. Now once these get entry inside the once they get in through the nose or mouth, these get entry inside the cells, host cells. So how do they enter? Since they have spike proteins on their envelope, those spike proteins bind with the ACE2 receptors on the respiratory epithelial cells, most commonly the type 2 alveolar cells. Okay, most commonly to the type 2 alveolar cells. Why? Because type 2 alveolar cells most commonly contain the ACE2 receptors. That's why the spike proteins mostly bind to the type 2 alveolar cells via their ACE2 receptors. So after binding to that ACE2 receptor of the type 2 alveolar cells, they get entry inside the type 2 alveolar cells. Now we know that the type 2 alveolar cells are have the function of producing the surfactant. And what is the function of the surfactant that we have read in physiology? The surfactant uh, causes or decreases the surface tension. Okay, surfactant decreases the surface tension. Now due to as these type 2 alveolar cells are infected, so the synthesis of surfactant is reduced. The synthesis of surfactant is reduced. As a result, the alveoli tend to collapse. As a result, the alveoli tend to collapse because the surface tension now increases. Due to decreased surfactant, the surface tension increases, and as a result the alveoli tend to collapse. To prevent this, inspiration becomes hyperactive, because if there is already any collapsing structure in the lung, then the body will try to fill it with the air so that it does not collapse. That's why the inspiration becomes hyperactive, so that the alveoli do not collapse. So that leads to increased interstitial space, because there is generation of the and once there is increased interstitial space, the as the interstitial space increases, the low pressure area is generated. Due to that low pressure, the fluid from the vessels move to the interstitial spaces, because the fluid moves from the high pressure zone to the low pressure zone. So the fluid moves from the vessels to the interstitial space, and that leads to formation of edema. This is edema. That leads to formation of edema. That leads to edema. And as edema develops, the SARS-CoV-2 in the lung, in the fluid that causes production of various cytokines, and excess amount of cytokines, different interleukins and other interferons, they together cause cytokine storm. And once the cytokine storm sets in, that causes the recruitment of the inflammatory cells, and severe inflammation starts. That inflammation causes lung injury, and as we have learned in the pathology, that any injury is healed by fibrosis. So here also, after the lung injury by the inflammatory cells which has been recruited due to the cytokine storm, and though that cytokine storm was due to the SARS-CoV. So after the lung injury there occurs fibrosis, and that when the fibrosis occurs, then the diffusion of oxygen decreases. That leads to acute respiratory distress syndrome, and finally, when it is reduced to a very large extent, the diffusion of O2 is reduced to a large extent, that leads to the respiratory failure. That leads to respiratory failure. Now, in that point, we have also seen the COVID-19, the earlier presenting the features of the COVID-19 disease are pharyngitis. Now we have to know why the pharyngitis occurs. So pharyngitis is due to, again, due to the ACE2 receptors which are present in high concentration on the pharyngeal epithelium. So as these ACE2 receptors are also present on the pharyngeal epithelium in a very high concentration, that's why the virus also infects this pharyngeal epithelium, and hence there occurs pharyngitis. And that's why we see the pharyngitis as the early manifestation in COVID-19. So what we have read in the pathogenesis, the virus gets entry via the ACE2 receptors. The virus gets entry via the ACE2 receptors, and that leads to decreased synthesis of the surfactant. After decreased synthesis of surfactant, the surface tension increases. The alveoli hence tends to collapse. Now, to prevent the collapse, the body increases filling the alveoli with air. Now, to fill the alveoli with air, the body has to increase the inspiratory rate. That's why the inspiration becomes hyperactive, and as the inspiration becomes hyperactive, the interstitial space increases. The interstitial space gets increased, and when the volume of the interstitial space increases, the pressure in that interstitial space decreases, leading to diffusion of fluid from higher pressure area to the lower pressure area which is present in the interstitial space. The fluid accumulates in the interstitial space, that leads to edema. And once edema occurs, that is the pneumonia like condition occurs. So after that, the SARS-CoV causes release of the cytokines, leading to cytokine storm. So once cytokine storm occurs, that leads to the recruitment of inflammatory cells. So after the cytokine storm, the inflammatory cells are recruited due to those interleukins, and inflammatory cells cause damage to the lung, and which is healed by fibrosis. And continuous fibrosis decreases the diffusion of the oxygen across the alveoli. As a result, after a long time, there develops respiratory failure. Now coming to the lab diagnosis part. So in lab diagnosis we have specimen collection. In lab diagnosis we have specimen collection, the first part. In any lab diagnosis cases, we have to first see the specimen collection. So here also you'll see the specimen collection as the first part. So the preferred specimens in case of the COVID-19 disease are the throat swab, nasal swab. Yeah, these two are the preferred specimens in case of the COVID-19 disease, and these are collected by Dacron swab most commonly or most preferably. Why we don't use the cotton swab? Because that may cause damage to the structures of the virus. That's why we do not use the cotton swab, and we prefer the Dacron swab. Now coming to the procedure. So in the diagnosis of the COVID-19 disease, the procedure is very much important to prevent the spread of the disease. So procedure should be done. The procedure that is given by the guidelines of India should always be followed, and the specimen should always be collected taking the full precautions, like wearing the PPE, gloves, face shield, etc., which are the component of the PPE. So wearing all these things, we should collect the specimen, not without them. Now, transport of specimen also plays a very important role. in lab diagnosis, because mm, transport in a unfavorable condition may lead to negative results that may be harmful for the society. So transport of a specimen is also very important. So how do we transport? We take the swab and they are dipped into the viral transport media, which is called as the VD. And these are some of the conical facts uh, with some liquid material, or in other words the liquid media inside them. And so these swabs, after collecting, are dipped into the viral transport media, and then they are packed into the triple layer pack, and then transported to the lab in a cold chain, storing at 4 degrees centigrade for less than 5 days. So we can store for up to 5 days at 4 degrees centigrade. If we want to store the specimen for more than 5 days, we have to store it at still at a much lower temperature, like less than 4 degrees C. Okay, so generally we keep it at 4 degrees C, and we uh, uh, transport the specimen to the laboratory within 5 days. Now the step that is done in the laboratory is the NAT test, which is the nucleic acid amplification test, of which the test is the real-time RT-PCR. That is nothing but a nucleic acid amplification test. The real-time RT-PCR is a nucleic acid amplification test. This is the gold standard method for the diagnosis of the COVID-19 disease. So, in as we have read in biochem, that RT-PCR is always, in RT-PCR we have to amplify the target genes, and then um, and detect those target genes. So here also we have certain target genes in the SARS-CoV-2, which we detect via this RT-PCR. Those target genes are the spike protein S, which is S gene, envelope protein E gene, and the membrane protein M gene. How do we remember these gene targets? We can remember it by SEM. Okay, so SEM genes are the target genes for the diagnosis of the COVID-19 by the real-time RT-PCR. What is the procedure of this detecting this target genes? So, as in all the PCR polymerase chain reactions, we have to first amplify the target genes. So, target genes are amplified first, then probe with the corresponding genetic sequence, with the corresponding nucleic acid sequence, and tagged with some fluorescent dye. They are left to bind with those amplified target genes. So the probes with the fluorescent dye, they bind to the target genes, and when they bind in a sufficient amount, that leads to generation of the fluorescence. So the point of cycle at which the fluorescence is generated is called the threshold run. Okay, so if the fluorescence is generated at within 38 cycles, within 38 cycles, then we call it as the positive test, because there is sufficient amount of gene, and within 38 cycle the fluorescence is generated. That means the test is positive. If it is generated at suppose 42, after 42 cycles of amplification, then the test is supposed to be negative. Okay, so that's how we calculate it, or we report the sample as negative or positive. So see here, the cycle threshold of run is the point of cycle at which the fluorescence is generated, and sample is considered positive only if the CT value is less than 40 cycles. So if the fluorescence is generated within 40 cycles, that means the sample is positive for COVID-19. 19. If you have seen the COVID-19 test reports, then you know that there is given CT value. So if those CT values are less than 40, that means the sample is positive. The next method, which is based on the nucleic acid amplification test, NAAT, is the automated real-time RTPCR, that is available as TrueNet and CBNet Gene Expert, which is nothing but the same machines which are used in the detection of TB. So for using CBNet, the target genes are the for using CBNet, the target genes are the E gene, that is for screening. The target genes are the E genes for screening, which is called as screening gene, and antigen, that is called as the confirmatory gene for SARS-CoV-2. Screening means if the E gene is detected, that means that particular virus belongs to the beta coronavirus genus. Okay, so if the E gene is detected, that only means that the virus belongs to the beta coronavirus genus, but does not confirm the SARS-CoV-2. For confirming the SARS-CoV-2, we have to detect the N2 gene. So once the N2 gene, which is the confirmatory gene, is detected, that means it is nothing but the SARS-CoV-2 virus. What is the advantage of this automated real-time PCR by TrueNet and the CBNet or Gene Expert over the real-time RT-PCR? This is the gold standard method. The advantage is that these machines have widespread availability even in the primary healthcare centers. These TrueNet and CBNet are found. The other advantage is that the turnaround time is very less. The turnaround time in TrueNet and CBNet is very less. These are fully automated. Minimum human handling is there. Minimal human handling occurs in this automated real-time RT-PCR machines. So these are much better than the real-time RT-PCR methods. Next is the antigen detection. So after collection of the nasal swabs, these nasal swabs are immersed in the viral extraction buffer. What is the use of immersing that nasal swab into the viral extraction buffer? The use is that after the immersion into the viral extraction buffer, the antigens are released, and then we can detect the antigens. Now coming to the antibody detection part. The antibody detection. So against any infection, we know that after certain time duration, there is formation of antibodies against that infection. So here also we find the formation of antibodies, like IgG antibodies are produced after 2 weeks of onset of infection. So as it is produced after 2 weeks, so the diagnostic use is nil. There is no diagnostic role of antibody detection in diagnosis of the COVID-19, but yes, it can be used for serosurveillance purposes. What is that serosurveillance? Serosurveillance means to know how many people of a community are already infected with a virus. So to detect the uh, proportion of already infected people in a community, we can use this antibody detection method. This is called as serosurveillance. And why do we want to do the serosurveillance? So the purpose of doing the serosurveillance is that to make proper policies, so that we can further control the infection from spreading to the other people. So for making the proper policies and good policies for the community, we want to do the zero surveillance, and that is done with the antibody detection. The next way of detection is the sequencing. Sequencing. So this sequencing nothing but helps in the detection of the mutations, and by this sequencing method we can detect the newer strands of the COVID-19, the newer strains of the SARS-CoV-2. Okay, the newer strains can be detected by the method of sequencing. Other than that, we have the viral culture method that is not recommended for diagnostic purpose, rather used for the research purposes, as we have read earlier as well, that viral culture is recommended for the research purposes only. Now vaccines of the coming to the vaccines of the COVID-19. So there are broadly three types of vaccines developed through in the entire world. There may be some other types of vaccines also, but these are in light. These types are mostly in the light, and these are the inactivated virus based vaccine, the viral vector based vaccine, and the RNA based vaccine. The inactivated virus based vaccines are the Covaxin, that has been developed in India indigenously by the Bharat Biotech, Sinopharm, and the CoronaVac. These are the Chinese vaccines. So these are the inactivated virus based vaccines, which are Covaxin, Sinopharm, and CoronaVac. These are the inactivated virus based vaccines, which out of which the Covaxin is developed by Bharat Biotech and is indigenous to the India. Now coming to the viral vector based vaccines, these are the Covishield, that has been developed by joint efforts of the Oxford AstraZeneca and the Serum Institute of India, Pune. So in India, two vaccines have been developed, which are the Covaxin and the Covishield. Covaxin and the Covishield. While other viral vector-based vaccines are the Sputnik V, that has been developed in Russia, Johnson and Johnson, which is the only vaccine allowed in a single dose. Okay, all other vaccines are given in double dose, but this Johnson and Johnson is the only vaccine which is given in a single dose. Just single dose is enough. Okay, that is Johnson and Johnson, and is a viral vector based vaccine. And which vector is used? That is the adenovirus vector we use for development of these vaccines. The RNA based vaccines are the Pfizer by the BioNTech Pharmaceuticals and the Moderna. These two are the RNA based vaccines developed in the foreign, and these are the RNA based vaccines. So these are all the vaccines which have been developed and currently in use in India. Many of them have got approval, and the vaccines which are indigenous to India are the Covaxin and the Covaxin and the Covishield, which are indigenous to India. India has developed by its own these two vaccines, out of which the Covishield has been produced by the joint efforts of the Oxford AstraZeneca and the Serum Institute of India, Pune, while Covaxin indigenously by the Bharat Biotech, without help of any foreign university or any other country. Sputnik V from Russia, this has also been approved in India. Johnson and Johnson has also got approval. So these are all the vaccines which are allowed or which has got approval in India and are in use nowadays. So this is all about the COVID-19 disease. This is a very important topic for upcoming university exams, because this is a very recent topic. So this is all about COVID-19.